Today we're going to take you on a little bit of a journey of what we do every day. Just go out and go to that. Hi, so I'm Dane Hazelbaker at Biopharma, and today I'm going to talk to you about what you can do if you want to become a gene editor like me. So gene editing is very exciting technology that's going on right now. Actually, the Nobel Prize in 2020 was just won by two outstanding scientists, Jennifer Doudna and Emmanuel Charpentier, for their discoveries in how to apply genome editing to human cells. So the cool thing about gene editing is you can dive into the human genome. There's 6.2 billion bases or letters per human cell. If you were to try to read all these bases, it would take you 10 years non-stop to do. However, with today's technology, we can quickly read and edit these bases. So for example, if you want to edit a single base out of these 6.2 billion bases, we can use editing enzymes, such as CRISPR-Cas9 technology, to go in, alter the DNA by physically breaking it, and then allowing DNA repair, either directed by us or through the cell, to change that base to the desired edit. What this can enable is not only a basic understanding of biology and medicine, also agricultural progress, diagnostics for diseases, and creation of therapies for genetic disorders, and even therapies that can enable cancer therapies, which is what we do more about. Okay, so now we're going to take a look at some of our DNA sequencing that we use to not only read the genome, but understand the type of edits that we make. We do this sequencing not only to confirm the editing we desire has occurred, but also we check other areas of the genome to make sure editing hasn't occurred in places. So this is really cool technology. Come on with me. So we keep a lot of our equipment confined in a climate-controlled room. And so what you see here is Gabby is running a DNA sequencer and checking her results. Now in this particular sequencer, she can read up to 7.5 billion bases in one day. And just behind her is an even larger sequencer that allows you to read 330 billion bases, all in the course of about 2 days. Now, if you remember, if you were to read these bases yourself, this would take you well over 100 years to do. Other equipment we have has to deal with confirming the quality of our samples and understanding their characteristics prior to sequencing. And after the sequencing, the data comes off in file format that then is run through statistical and computational analysis to understand the nature of the changes to the human genome. So that, in a nutshell, is what we do day to day as gene editors. Now, if you're interested in gene editing, I would recommend in school focus on biology, chemistry, math, in particular statistics, computer science. As you see, we use computers for almost all aspects of analyzing our editing experiments. And also, arts and humanities are very important. I believe that biology is our way of understanding the beauty of nature, and so understanding the beauty elsewhere really helps in education towards something creative. And one thing I love about genome editing is we get to be the Indiana Jones of the genome.
Hi, I'm Gabby. I'm a senior associate scientist here at Biopharma. At Biopharma, we're using genetically engineered cells to help treat cancer patients. In the lab, after we genetically engineered cells, we need to learn about what happened to them at a DNA level. So that's where I come in. So a lot of what I do is editing the cells and then sequencing their DNA. But there's a lot of steps that happen in between that. So to sequence DNA, you have to do a lot of liquid handling. So one of the coolest parts about being a scientist is that we get to use new and up-and-coming technologies. So when I have to move liquids around to be able to prepare my samples for DNA sequencing, traditionally I would use a manual pipette like I have here, and I would move this blue fluid into a plate only 12 wells at a time. Now imagine having to do that thousands of times just to be able to figure out what you did in the lab. To help solve this problem, we're able to use robotics and all sorts of cool engineering to do this step for us so we can have it be done faster, more accurate, and saves me as a scientist a lot of time. This is a liquid handling robotics system, and pretty much what it does is do that pipetting of liquid for me. If we run this instrument, you can learn about how smart it is and how much technology really went into it. So now that we've started the instrument, it's first going to float around and take a look at what we have in there and make sure it matches what we told the computer it has. So this is essentially just making sure it has all of its supplies before starting, like you would if you were baking a cake. You wouldn't bake the cake if you didn't have eggs. Right now, it's counting how many of each of these things we have, and also it has the ability to check all sorts of volumes. This instrument is really awesome to save a lot of time. It's very accurate and it's easy to use. So I am not a robotics engineer, so this really helps us, and it gives scientists certain niches to specialize in. I love finding out all of the things that you can't see. I think that's really exciting. I love my job because it's different every day. It's active. Some days are really crazy and other days are more low-key and they're a lot of fun. So your job, it changes every day. Growing up, I was always interested in nature and animals and the natural world, and this is how I can look at it at a microscopic level. So often I can't see what I do every day, but I get to use all these cool tools and different new cutting edge technologies to figure out what we did and to see the things that you can't see.
Hi, I'm Jess, and I'm a scientist here at Robotic. More specifically, I'm a cellular biologist. Cellular biologists are scientists who study cells, their structure, their function, their behavior, and how they interact with other cells. Now that Dane has designed the desired edit, and Gabby has edited the cells, they give the cells to me so that I can study them. To do this, we must first grow the cells in vitro. This means that we place the cells in a plastic container or a flask with the appropriate food called media, which gives them all the nutrients they need to survive and grow. On a daily basis, I feed and maintain the growth of these cells. As a cellular biologist, there are many procedures called assays that we use to study cells. One of the key assays I use is called Photocytometry, using a machine called a Photocytometer. Photocytometry involves labeling the proteins that sit on the cell's surface with fluorescent antibodies. In a complex mixture of different cell types, we can distinguish individual cells based on their fluorescent antibody tags, and these are detected by the lasers of the Photocytometer. You can also study cells by looking at them under a microscope, as shown here. Different cell types have different morphologies, or shape and size under a microscope, and we can even watch cells move or migrate under the microscope as well. What I love about my job as a scientist in general is that I get to discover new things every day. I get to problem solve. I get to use cool new technologies. What I love about being a scientist in industry in particular is that we get to work together as a team to reach a common goal. And further, I love that our research can make a direct impact by creating new treatments that can improve patient lives. Another aspect is that you get to travel and you have the freedom to do science anywhere. Originally, I'm from Australia, where I did all my scientific training. That's what I love about science and being in industry, is you can do it anywhere. At high school, I went and did a double degree in law and applied science. However, halfway through the degree, I decided that I loved the science units more, so I dropped the law component and completed a degree in applied science. After dropping the law component, I thought, what actually can I do with a science degree? So a lecturer suggested I do a small research project during my undergraduate. With that small undergrad research project, I decided that I love research so much that I wanted to do it for my career. So I went to grad school and I got my PhD. However, you don't need a PhD, and therefore associate scientists aren't required to have a PhD, only an undergraduate science degree. So in summary, my advice is to do an undergraduate science degree in cellular and molecular biology. And whilst you're doing your undergraduate degree, I would suggest to do a co-op or an internship to gain some industry experience and to also see what type of research you'd like to go into in the future. Thank you. I hope this has been informative, and I hope you now understand the myriad of jobs that you can do as a scientist in industry.